The antibody responses are driven by CD4 T cells of the phenotype called Th2. Please take a look at my other video on the Th phenotypes. When we are encountered by a danger signal, which in this case I'm putting here boy bitten by lizard by Caravaggio. Also by Caravaggio, I'm putting this basket of fruit because both the venom or the basket of fruit can actually trigger a lot of antibody responses. Let's say in this basket we could have ants in there that have some venom if we touch them and they bite us, or you can have food allergies, or you can have pollens in there. In addition, we can have infections, like in this case the self-portrait by Edvard Munch when he had the flu. So there are many different triggers that are danger signals that will be identified by our dendritic cells. They travel through the lymphoid vessels to the lymph nodes. And it is the dendritic cell that carries the piece of the danger signal, which here is the red oval. And that is the one that the T cell can see. This is presented via the class 2 of the major histocompatibility complex molecule, because this class 2 will be identifying the CD4 T cell. And it's the CD4 T cell, the one that drives the antibody production. Now, in addition to the presentation of the antigen, we need what we call the co-stimulation, which I always like to say, it's like a handshake. It's like the dendritic cell needs to have this double signal system to the T cell so that the T cell actually responds. If they only present the antigen without that handshake, then the T cell does not respond. And this handshake is very, very important. Like the dendritic cell is reaching out to the T cell. This is true for many other T cell activations. And if you realize, I have put a different kind of handshake. Each one is different, and each one of these handshakes are going to trigger a different response in the T cells. So in this case, we're going to be producing interleukin-4, and that is going to differentiate the T cells in this lymph node as Th2 cells. They proliferate, and this is what makes the lymph nodes grow. Oftentimes, if we have an infection or if there's a cancer, we can touch those lymph nodes and they are swollen. They're also painful. That's when the T cells have identified a danger signal. They are specialized to identify it and they proliferate. But they really don't help us if they stay in the lymph nodes. They need to reach the place where the danger signal is. So they travel back through the lymphatic system, through the blood, to the tissue, either that has been where the venom was injected, or if we ingested it in the GI system, wherever this danger signal is. This T cell has been given a fingerprint of the whole danger signal by the dendritic cell. It's like the dendritic cell provided the fingerprints of the culprit. And now the T cell can identify the individual just by using the fingerprints. However, the Th2 cells are not effector cells. They cannot really do anything to the danger signal, but what they do is that they actually stimulate other cells, and the activated B cell, directly a consequence of this Th2 response, will then differentiate into plasma cells, and it's the plasma cells, the ones that will produce antibodies directed specifically to that danger signal, and that's how we deal with the danger signal using antibodies. Let's begin reviewing the primary antibody response. So for now, let's forget the memory cells. When we have the challenge, which is the first time that a signal is perceived as a dangerous signal, the naive B cell will differentiate into the activated B cells. These activated B cells were driven by CD4 Th2. cells and signaling, they are specifically identifying the danger signal. They differentiate into plasma cells that are the ones responsible for the production of antibodies. And what we have first is the short-lived plasma cells. This process takes anywhere between 5 and 15 days, depends on the danger signal, and it also depends on the immunological status of the individual. How good is the immune response of the individual when they encounter this danger? I want to also mention that there's an exception to this timeline. The very fast response that occurs with allergic or anaphylactic reactions that can be within minutes, and it's characterized by IgE, is covered in a different tutorial in YouTube. Besides the anaphylactic responses, the other B cell response, as I mentioned, takes about a week or so to have the titers detectable. In the y-axis is the amount of antibodies shown in this hypothetical graph, where the blue curve is how the antibodies start rising until they get to the peak. When they get to the peak in this primary response, it's basically IgM. These are the product of these short-lived plasma cells, and they all identify the danger signal. A few of them could be IgG. If you want to look at the isotype switch video I have, I'm also adding a link to that video. What happens over time if the individual is not exposed to that same danger signal is that the titers of the antibodies now are going to go down, and it's basically because there are some long-lived plasma cells in the bone marrow. These were triggered by the same initial danger signal. That in time, if there's no further exposure to the danger signal, they will eventually disappear and the titers will go back to zero. That is the primary antibody response. The secondary antibody response is characterized by having memory B cells. What this means is that the B cells have created memory against the original danger signal. So when we encounter the same danger signal or something similar, our B cells are very quickly able to differentiate into plasma cells and produce very high levels of antibodies to the danger signal. These are now IgGs because they have matured and they can effectively deal with the danger signal. Like if it is an infectious agent, they can do this. Now, the characteristic of this secondary response is not only that there's very high titers, but it responds very quickly to this re-challenge. So this characteristic of the secondary response that is very fast after re-challenge, very high titers, and these are IgG antibodies to the danger signal, is a protection that our body gives us against danger. Michel de Notre Dame, also known as Nostradamus, survived a plague that killed his family, many of his friends. All of his children died. And he was known to have written that he did not want to use any more protection when he treated patients because he did not care if he died. However, he survived and he kept treating patients throughout his life that had the plague, and he never actually got sick with the Yersinia pestis. And this is most likely because he did have memory. He had built a very good immune system protection with memory against this very dangerous bacteria. Examples of memory that we can easily think about is what we trigger with vaccinations. Now, the memory B cells also can produce diseases like allergic diseases, and this memory will vary. It varies how long it stays with us. In some cases, it's very long-standing, and in some cases, it disappears. And if it's a protective antibody, we may need to have booster immunizations. If it's an allergy, people might say, I used to be allergic, I used to have asthma, or I used to have some sort of rhinitis, and it disappeared. So that can happen in some cases, and it depends on the individual, and it also depends on the kind of signal. But the memory B cells that could be created oftentimes do not behave exactly the same way in the same individual for all the different danger signals. I hope you enjoyed this video and consider subscribing to my channel, and please watch other videos in my own channel. Thank you very much.